Hi all, I hope you are doing well. I'm doing okay as well, so I hope everyone else is just staying at home and doing well. So today we will be talking about the next module. I'm starting DNA repair today, so DNA damage and repair. I'm going to post two different set of slides today so that the video is not too long. The topics that we are going to cover today is type of mutations, mismatch repair, types of DNA damage, and I might not able to get to direct repair, but if we do, that will be another topic. So we are following chapter 10 now. This part of the slides are from page number 313 to 326 of Watson 7th edition. All right, so before I go into DNA damage and repair, I just wanted to make sure that everyone is on the what we're talking about. So we talked about DNA replication, and if you think about a DNA replication, it's really all is the part of the cell cycle. So cell cycle goes into different phases. Interphase is that's where the DNA, the cell prepare itself for division, and mitosis is when it actually divides. Mitosis, that's where chromosomes will divide, and then cytogenesis when the cell will divide into two. In the G1 phase, most of the cellular contents, excluding the chromosomes, are duplicated. In S or synthetic phase, that's where the chromosomes are duplicated. So when what we are talking about DNA replication, we are talking about the S phase of cell cycle. In G2 phase, the cell double checks the duplicated chromosomes, make sure that there is no error. However, this checking is also going on along with the synthesis as well. So as the DNA is being synthesized, being replicated, the polymerase will keep checking if there is anything that's wrong with the DNA molecule, with the new addition of nucleotide. But in G2, everything is double checked to make sure there is no changes, no mutations. But mutations still happen, and those. But a DNA damage is not a mutation until it went through the repair. If the repair cannot detect it, or it goes undetected by the DNA repair mechanism, then it's a mutation mechanism. Checks everything and tries to repair all the errors. And once that happens, then the mitosis, then it goes into the next phase, mitosis phase, and the DNA and the chromosomes divide. Right, so we will be focusing more on, so we already focused a lot on the synthetic phase, S phase. Now we are talking about synthesis as well as G2 phase of the cell cycle. Okay, so what is a mutation? A mutation is a permanent change in sequence of DNA. It usually happens by the activities of DNA interacting proteins, so replication errors lead to mutation. Unless it is detected, right, by the repair system. There are different types of mutations, and you've learned about these already. There could be point mutation. Could be a point mutation is single nucleotide, usually insertions and deletions, or indels in short. Indels can be point mutation, or it can involve more than one nucleotide insertion or deletion. And microsatellites, these are the hot spots of mutation. That's where because these are the repeat sequences, very short repeats, the mutation tend to occur in microsatellites as well. Point mutations involve the change. This change can be a transition or a transversion of a single nucleotide. Okay, this can be a single nucleotide insertion or deletion. Transition is pyrimidine to pyrimidine or purine to pyrimidine, purine to purine substitution. So it's not changing the type of your base. It's always from either pyrimidine to pyrimidine, so that will be cytosine and thiamine, and purine to purine, A or G. Transversion is the opposite, so it's purine to pyrimidine or pyrimidine to purine. So here as an example, so this will be your transition. That's where either these two purines, these can interchange, or these pyrimidines can interchange. They can substitute each other. So that will be a transition mutation. Transversion is when it's between the purine to pyrimidine and pyrimidine to purine. Common mechanism for mutations is replication error. So although DNA polymerase proofreads, but proofreading is not 100% accurate, it can still slip through, especially the transitions can slip through the proofreading ability of the DNA pol. Changes in structure of DNA, that means a damage to DNA that could be due to UV radiations or other substances that are DNA damaging substances, or transposition activity, that means transposons can enter or insert themselves in the DNA sequence that didn't have that sequence originally, and that can lead to mutation as well. Some base analogs can also cause mispairing. For example, fibromuridine prefers to bind a G instead of an A. So these kind of errors can happen in the DNA sequence that leads to mutation. So here, this shows how mutations might happen. As this is first round of replication, this is what it's supposed to be. There is a misincorporation. So instead of a T here, a G has been inserted instead with A. But most of the time this can be repaired, and that the cell will repair it, and here is the repaired product. So this repair has happened and everything is fine. However, if this doesn't repair itself, then what will happen is then one of the sequence will be changed, because this A maybe in second replication led to T, that's correct, but where there was a G that leads to the corresponding nucleotide for that G will be a C. So here there's a point mutation has occurred. Insertions or deletions or indels, these can be single or multinucleotide events, and we have talked about these multinucleotide events, especially when we were talking about CRISPR, how a double-stranded DNA break can lead to multinucleotide insertions or deletions. This could be single, but usually these are more than one nucleotide insertions or deletions. The point insertions, when happen in a coding sequence, they can lead to frame shift. So here in this example, this is a wild type sequence. This codes for this protein that has methionine, lysine, phenylalanine, and glycine, and then a stop codon. But if a mutation happen in it, for example here there is an extra U inserted after AUG, what will happen is that the codon will be read as UAA, which is a stop codon, so that will lead to a frame shift. It's led to a frame shift and inserted a stop codon here. So this is a nonsense mutation. Now, frame shift can also cause extensive missense mutations. For example, in this part here, this missing U led to a frame shift as well, and that changed all the sequence downstream of that deletion. So the protein will be very different from what it meant to be originally, and it will not be functional because the sequence has changed now. When there is an insertion or deletion of 3 nucleotides, then there might not be a frame shift, because that will be a one whole codon that leads to a missing nucleotide if it's a deletion of 3 of a complete codon, or if it's an addition of one extra amino acid if it's an addition of one whole codon. So that these type of insertions or deletions could be harmful if that amino acid is really important, but they're less harmful than the extensive missense mutations or nonsense mutations. These longer insertions or deletions, they tend to disrupt open reading frame, because open reading frame is your coding sequence region, and that will, once that changes, then the protein sequence will change as well. So transposition, so transposable elements inserting themselves and strand breaks in DNA double-stranded break, for example, are usually results in these longer insertions or deletion instead of point mutations. Microsatellites, these are called hotspots of mutations. These are highly repeated short sequences, and because these are highly repeated short sequences, these are prone to mutations. So sometimes they expand, sometimes they contract, during replication or during recombination. So here, if you look at these microsatellite marker, this is a duplet of the C and A number of times it's present in the sequence, and this sequence can lead to, because DNA polymerase is not very good at understanding how or knowing how many sequences are there. It may insert or may delete the sequences of these microsatellites. These microsatellites, as I already mentioned in the class, these are also used for DNA fingerprinting, because different people have different numbers of the same microsatellite. So these marker number of these, how many times these repeats are in a genome, in an individual genome varies. Okay, so how to correct a replication error? So say, for example, we have point mutations or deamination. We haven't talked about deamination, so let's talk about deamination. So deamination is hydrolysis of cytosine into uracil. I think we talked about this previously in the class. The mutations usually do not block replication or transcription, but they can lead to a nonsense or missense mutation. So that means appearance of a stop codon in the coding region or change in the codon. So, but the DNA replication and transcription still occur. What it does, it changes the protein. It changes the protein in a way that either it adds a stop codon, so it's a truncated protein, shorter protein version than it was supposed to be, or it's a missense. That means there's a change in codon. That means it's a wherever the change has happened, after that change, after that mutation, the codon will change, or the protein sequence will change. Deamination is hydrolysis of cytosine into uracil. So when cytosine hydrolyzed into uracil, instead of that cytosine binding to a guanine, the uracil will bind to an adenine. So that will lead to a change. Or this uracil will be, because it's a U, so in mRNA sequence that will be read differently than it would have been as a C. Most transition and transversions are silent, so that means these are silent mutation. They have no effect on coding region or phenotype. However, these if these are not silent, then this can affect the protein sequence. So these point mutations, because we're talking about damaged and DNA now and DNA repair, just keep in mind that a point mutation is not a damaged DNA per se, but they may represent damaged or corrupted information. So these point mutations, until these are fixed in an organism, these can be repaired. As we all know, the DNA pol has proofreading capabilities. So DNA pol has this different subunit that checks the DNA, proofreads to make sure that all the nucleotides are correct, the incoming nucleotide is correct. But this 3 to 5 prime exonuclease proofreading activity is not foolproof. It can sometimes it misses some of the nucleotides that it cannot detect. So the mechanism for detecting these mismatches and repairing is called a mismatch repair. Mismatch repair raises the fidelity of replication number of folds 2 to 3 orders of magnitude. So when DNA pol makes an error, it's usually one error in about 10 to the power 6 nucleotides. But once the mismatch repair happens, this error is much fewer or much lower than it would be without the repair. So how does this mismatch repair works? As In E. coli, mismatches are detected by a dimer of mismatch repair protein called MUT-S. This MUT-S, as I said, these are a dimer of proteins, so two subunits come together to dimerize. These MUT-S, once they scan the DNA, once they recognize the mismatch, then they recognize the mismatch because of the distortion of the DNA strands. When DNA is damaged, it's easy to get distorted because it doesn't have the proper base pairing. This MUT-S, when it finds the problem from the nucleotide that's incorrect, it will stop there. It will bind to that nucleotide, and this complex of MUT-S and mismatch carrying DNA will recruit a second protein called MUT-L. This MUT-L then activates or recruits another protein, which is an endonuclease enzyme called MUT-H. So this is the complete mechanism, and then I split it into two here so that you can see it a little better. So here there's distortion in DNA because this is an incorrect base pairing. It's T to G. It shouldn't be T and G, right? It should be either C or G, or it should be T and A. So what kind of mutation, what kind of change it is if one of these is correct? Say for example, if this is G and here it's supposed to be C, but it's a T, it's a transition, right? Because the C and T, they're both pyrimidines, and instead of C there is a T. Anyway, so this MUT-S, this dimer, these two proteins, these are scanning the DNA molecule. It finds this problem, this incorrect pairing in the DNA, and it stops there. It holds on to this wrongly paired nucleic acid, and it stops there. Once it stops there and finds this problem, it will recruit MUT-L. This MUT-L will then recruit MUT-H. MUT-H is an endonuclease. This MUT-L will bring this endonuclease to the DNA molecule where MUT-S is bound, and this endonuclease MUT-H will make a cut in the DNA molecule in one of the strands. Okay. Then a special helicase, UVRD. So UVRD opens the DNA molecule here, the two strands of DNA, and an exonuclease will make these, will remove more nucleotides and get to the point where there is a problem. Once this has been removed, then this DNA polymerase, just like the DNA replication, will fill this 5 to 3 prime, in the 5 to 3 prime direction, fill the gap. Mismatch has been repaired. Ligase will come and fix the final nick. So this is how it works in E. coli. So again, MUT-S scans the DNA, the complex of MUT-S and the mismatch carrying DNA recruits MUT-L. MUT-L activates MUT-H. This is an endonuclease that causes the single-stranded break in DNA near the mismatch site. This nicking is followed by the specific helicase UVRD and one of the 3 exonucleases. These exonucleases then remove the portion of the DNA leading to including the portion that is mismatched, and the resulting gap is filled with DNA pol 3 and ligase, right? Just like the DNA replication. But how does the E. coli cell knows which of the 2 mismatched nucleotide to be removed? Remember in this example here, if we go back to this slide, so this could be either of these, could be the incorrect one, the T or G, right? But how does E. coli knows which one to cut? And E. coli or bacteria in general have this really nice system. They have this special enzyme called DAM methylase. This DAM methylase adds methyl group to A residues on both strands of the sequence at 5' GATC 3'. So here, this is GATC. So it will add a methyl group on A. So this is the parental strand. The parental strand already has the methyl group. The daughter strand will get the methyl group once once the replication is going on. So demethylase works with the replication fork, but it's behind the mute H. So mute H, it's still looking at if there is any problem, and demethylase puts this methyl group in the daughter strand behind mute H. So that means for I'm getting ahead of myself. So this GATC sequence is widely distributed in the entire genome of E. coli. So every 256 base pair there is a GATC group, and all of these GATCs have adenine methylated, and all these methylated by demethylase. So when a replication fork passes through DNA, which is fully methylated at GATC, both strands, the resulting daughter strand will be hemimethylated, which means the daughter strand is not methylated at this point, so it's only one. So this is replication fork, right? So this parental DNA is methylated on both strands, but this parental DNA opens and daughter strand is forming here. So daughter strand has methylation only at one point, not on the daughter strand yet, only on the parental side. So it's hemimethylated at this point, right? So for a few seconds, up until demethylase catches up and methylates the daughter strand, there's only one template strand. Only the template strand is methylated. So the mute H knows which of these DNA strands to be cut, because it will be the one that's not methylated, so the daughter strand. So it keeps the sequence of whatever it was in the parental strand and fixes the one that would be in the daughter strand. So this is really good. This is very clever of E. coli to use this system. But vertebrates do not have, or eukaryotes do not have demethylates, so we do not use this very clever technique of E. coli to fix our repair system or fix our DNA. Okay, so this is what we talked about prokaryotes mismatch repair system so far. The repair system has the enzymes that are conserved in eukaryotes as well. They have different names, just like everything else. So here, the mismatch detection enzyme in prokaryotes is mutase, that we talked about, those dimer of proteins that bind to by scanning, S for scanning. So these are the mutase proteins. The orthologous to these mutase in eukaryotes is MLH and MSH. The proteins that activate endonuclease, mut-L in prokaryotes. In E. coli, the mut-L brings the mut-H to the site where mut-S is bound to the DNA that is incorrectly paired. In eukaryotes, this mut-L equivalent is pCNA and RFC. The endonuclease that cuts that single-stranded cut, makes it daughter cell, the cut on the daughter strand is called mut-H in prokaryotes. In eukaryotes, it's called XO1, but there probably are more endonucleases that we don't know yet. The helicase that opens up that part of DNA strands is called mut-H, and it's called UVRD, I'm sorry. In prokaryotes, there is no orthologous helicase in eukaryotes that has been found so far. And the exonuclease could be 5' exon 7 or 3' XO1, and we will talk about it, why there are two 5' or 3'. And you can think about it, if mut-S is bound towards the on one side of the, it depends on where mut-S or not mut-S but mut-L and H are bound on the DNA strand. Depending on their location, they can be on the 5' from the mismatch, or they can be on the 3' on the mismatch, and depending on where these are located, they will need the exonuclease that can chew up, leading to the incorrect base pair. In eukaryotes, the exonuclease is XO1, which also has an endonuclease activity, but we don't really know for sure yet. Strand replacement and finishing is carried out by DNA pol 3 and ligase, and in eukaryotes by DNA pol delta and ligase. So now I'm going to move on to the next set of slides. So I will be posting both of these videos today, this one and the next one.